Hello, I am Professor Sims, and in this video I will discuss genes and proteins. This is the fifth of the series of 10 lessons held as part of my general genetics course. If you're a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. The learning objectives for this unit include explaining the central dogma of DNA protein synthesis. It's the same as the central dogma for biology and genetics. Describe the genetic code, its role in amino acid and protein sequences. We're going to discuss the processes involved in prokaryotic and eukaryotic transcription and the significance of transcription factors, the different steps in RNA processing, the significance of exons, introns, and splicing for mRNA. We'll talk about how tRNAs and rRNAs are processed, and we'll describe the processes involved in protein synthesis. Since Mendel's work was rediscovered in 1900, our understanding of genes has evolved significantly. Genes are no longer an abstract concept. They're actually tangible molecules that are made of DNA. They're organized linearly on chromosomes, and they carry instructions for building proteins, which are crucial for cell functions and for life itself. At this point, we've heard quite a bit already about how DNA contains the genetic code, but more specifically, the genetic code refers to the DNA alphabet and the RNA alphabet and the polypeptide alphabet. So that's the sequence of amino acids. The genetic code comes to exist via the central dogma. We talked about the central dogma in the Intro to Genetics lecture. It consists of 3 steps: DNA replication, transcription, and translation. We discussed DNA replication in lesson 4, and today we're going to talk a lot more about transcription and translation. Very briefly, transcription is the process of creating messenger RNA copies of genes. These mobile copies contain an alphabet of A, C, G, or U, and the mRNA is then translated on ribosomes, converting the nucleotide-based information into proteins. This process of DNA protein synthesis, it follows a systematic flow of genetic information. So you may remember this figure from the Intro to Genetics lecture, right? It's the 3 steps. You've got your DNA replication, transcription into mRNA, and then translation into protein. Figure 15.3 shows these steps in another way. The DNA code has the instructions, right, which gets transcribed into messenger RNA. Ribosomes read the code from the mRNA and use it to make amino acids, which later fold to make proteins. There are thousands of proteins in the human body, and they're all made up from just 20 amino acids. These 20 amino acids are encoded for by 64 triplet codons. What we have is 64 groups of mRNA letters that are making the code for 20 amino acids. The use of multiple codons to represent the same amino acid is known as degeneracy. This redundance helps to buffer against the impact of random mutation. Codons for the same amino acid usually differ by only 1 nucleotide, and codons for chemically similar amino acids tend to share similarities. For example, aspartate and glutamate both fall in the GA block and have negative charges. So this aspect of the genetic code helps to ensure that a single nucleotide mutation might still specify the same or a very similar amino acid, and this prevents the protein from becoming totally non-functional. The genetic code is almost universal, with only a few exceptions. Nearly all organisms rely on the same genetic code for protein synthesis, and this widespread use of the code strongly suggests a common origin for all life on Earth. It's quite remarkable, considering the vast number of possible combinations between 20 amino acids and 64 codons. The Crick-Brenner et al. Experiment was a scientific experiment performed by Francis Crick, Sidney Brenner, Leslie Barnett, and R.J. Watts-Tobin. It was a key experiment in the development of what is now known as molecular biology, and it led to a publication entitled The General Nature of the Genetic Code for Proteins. The study demonstrated that the genetic code is made up of a series of 3 base pair codons, which code for individual amino acids. The experiment also elucidated the nature of gene expression and frame shift mutation. In the experiment, proflavin-induced mutations of the T4 bacteriophage gene were isolated. Proflavin causes mutations by inserting itself between DNA bases, resulting in an insertion or a deletion of a base pair. Through the use of proflavin, the experimenters were able to insert or delete base pairs into their sequence of interest, and when nucleotides were inserted or deleted, the gene would often be non-functional. However, if 3 base pairs were added or deleted, the gene would remain functional, and this proved that the genetic code uses a codon of 3 nucleotide bases that correspond to an amino acid. So this is figure 15.2, and it's showing the structures of all 20 amino acids. I don't want you to worry, you don't have to memorize these. I just wanted you to see that they all have the same amino group here and an acidic carboxyl group, and then they all have a different organic R group, or this side chain is what changes between the 20 different amino acids. Figure 15.4 is showing the arrangement of codons in the coding table, which reveals the structure of the genetic code. So there's 16 blocks of codons. Each are defined by the first and second nucleotides within the block. For instance, this is the AC block that represents threonine, right? Because all 4 of these code for threonine. Threonine is this guy right here. Some blocks are split into pyrimidine and purine halves, depending on the ending nucleotides. Certain amino acids have a full block of 4 codons, like alanine, threonine, the one we were just looking at, and proline. And then others have the pyrimidine or purine half of their block, like histidine and asparagine. Notice how histidine and asparagine have the last 2 codons are the same, but the first one is different. This is a C and this one's G. Some amino acids even have a block and a half, totaling 6 codons. An example of that would be serine, this guy here. He is encoded by these 4 codons, but also these 2 here. Again, you don't have to memorize this chart. This would be something that would be given as a resource if you needed it on an exam or a quiz. Oh, and I gotta talk about this codon here, AUG. He has a dual role. It codes for an amino acid, it codes for methionine, but it also acts as the start codon to kick off translation. So AUG is the translation start codon. Figure 15.5 is just showing how a frame shift mutation, we saw that in the last lesson, is essentially what the Crick and Brenner experiment discovered. They discovered that if you added or took away 1 or 2 base pairs, it would not only cause the amino acid at that site to change, but it changes all of the amino acids following that site because it shifts the whole reading frame over. Now, if you added 3 base pairs or you took away 3 base pairs, it would just remove this 1 amino acid, and then everything after that would still code the same as it would have before, right? So we've seen that before. Now that we have a good understanding of the genetic code itself, let's go into more detail about how it works. It is different in prokaryotes and eukaryotes, as are most things, so we'll discuss both processes individually. Prokaryotes don't have membrane enclosed nuclei, allowing transcription, translation, and mRNA degradation to all happen simultaneously in the cytoplasm. This efficient process rapidly increases the level of proteins through multiple transcription and translation events occurring on the same DNA template all at the same time. So prokaryotic genomes, they're compact and their transcripts often span multiple genes, resulting in what is known as polycistronic mRNAs that produce more than one type of protein. Transcription requires partial unwinding of the DNA helix. The gap that's formed in the process is called the transcription bubble. This is where the RNA nucleotides pair up with the nucleotides of the template strand to make a copy of the genetic code using the enzyme RNA polymerase. The specific nucleotide where the first 5' mRNA nucleotide is transcribed is called the +1 site, or the initiation site. Nucleotides before Before the initiation site are referred to as upstream, marked with a minus sign, while nucleotides after the initiation site are labeled downstream, or they have a plus sign. Figure 7 here is showing how RNA polymerase is adding nucleotides, and it does it in one direction. It does it in the 5' to 3' direction. You can see where the complementary copy of the DNA code is synthesized inside of the transcription bubble. That's this. This is the transcription bubble, and then this orangish thing around it is the polymerase, the enzyme. Remember that in prokaryotes, this is happening in the cytoplasm, and as the template is read, the DNA unwinds ahead of the polymerase and then rewinds behind it. That is, the bubble moves from the 5' to 3' direction as the genetic code is transcribed into mRNA. The RNA polymerase used by prokaryotes is the same for transcribing all of their genes. So for example, in E. coli, the polymerase consists of 5 subunits, with 2 being identical. And then 4 of the subunits, the alpha, there's 2 copies of alpha, and beta prime, form the core enzyme responsible for assembling on the DNA during transcription. The 5th subunit, it's called sigma, plays a role only in transcription initiation. It provides specificity, ensuring that the polymerase starts mRNA synthesis from the right initiation site, the +1 site. Without sigma, the core enzyme would produce nonsensical proteins, and the complete polymerase with all 5 subunits is called the holoenzyme. Promoters are crucial DNA sequences where the transcription machinery in RNA polymerase binds to initiate transcription. Promoters are often located upstream of the genes that they regulate. While promoter sequences vary among prokaryotic genomes, there are conserved elements. The upstream 10 and upstream 35 regions contain consensus sequences recognized by the sigma. The upstream 10 region has the TATAAT sequence, and the upstream 35 region has the TTGACA sequence. Interaction between sigma and these sequences allows the core enzyme subunits to bind, initiating the unwinding of the DNA template and production of abortive transcripts. Figure 15.8 shows how the sigma subunit of prokaryotic RNA polymerase recognizes the sequences found in the promoter region upstream of the transcription start site. That's the upstream 35, upstream 10, and then this is the +1 start site. After initiation, the next steps in transcription are elongation and termination. During the elongation phase, sigma dissociates from the polymerase, allowing the core enzyme to proceed along the DNA template and synthesize mRNA at a rate of about 40 nucleotides per second. So it's very fast in prokaryotes. As elongation progresses, the DNA is unwound ahead of the enzyme, rewound behind it, right? We saw that. To maintain stability during the mRNA synthesis, the RNA polymerase acts as like a stabilizer, a link between the DNA template and the new RNA strand. When a gene is transcribed, termination signals guide the dissociation of the polymerase from the DNA template and release the newly formed mRNA. These are the 2 types of termination signals, is row-dependent and row-independent. In row-dependent termination, you have the row protein that tracks behind the polymerase, and then near the end of the gene, the polymerase stalls at a G-rich region, and it allows the row to collide with it, releasing the mRNA. So it just kind of gets kicked off by the row protein. Row-independent termination relies on specific sequences in the DNA template. So as the polymerase approaches the end of the gene, a CG-rich region causes the mRNA to kind of fold back on itself, and it forms what they call a hairpin structure. And the hairpin stalls the polymerase when it transcribes at an AT-rich region, which leads to the detachment of the polymerase and the new mRNA transcript. Once termination occurs, the process of transcription in prokaryotes is complete. Interestingly, the prokaryotic transcripts are quickly utilized for protein synthesis because the transcription, translation, and mRNA degradation can happen simultaneously in prokaryotes. And this is because the absence of a membrane-bound nucleus allows them to happen all at the same time, on the same template, in the same direction. And that's what is being shown here in figure 15.9. So you can have proteins just forming all over the place at the same time on a prokaryote. In contrast, eukaryotic cells with their nuclei can't simultaneously perform transcription and translation, right? So we're going to talk about eukaryotes now. Both prokaryotes and eukaryotes share the fundamental process of transcription, but there's a few key distinctions. Eukaryotes have a nucleus and organelles, so that means that the mRNA has to be transported out of the nucleus and into the cytoplasm. And then we have to deal with protecting that mRNA so it doesn't get degraded before it can be translated. Eukaryotes employ 3 distinct polymerases, and they're each responsible for transcribing a specific set of genes. Eukaryotic mRNAs typically code for just one protein, making them monogenetic. In eukaryotes, transcription initiation requires the involvement of various proteins called transcription factors. These factors first bind to the promoter region and then assist in recruiting the appropriate polymerase to start transcription. Eukaryotic mRNA synthesis is considerably more complex compared to prokaryotes, and these transcription factors are kind of like air traffic controllers, right? They can regulate how genes are expressed. They control when and how much of a gene's instructions are read. It's like they have a key that fits into a lock in the DNA, and they can turn genes on and off. And this type of control is crucial for cell development, various biological processes, gene regulation, and even disease pathways. Table 15.1 is showing the differences in the different RNA polymerases in eukaryotes. RNA polymerase 1 is located in the nucleolus, a specialized substructure within the nucleus, and is responsible for transcribing, processing, and assembling ribosomal RNA, or rRNA, into ribosomes. rRNA, which constitutes a vital part of ribosomes, is not translated into protein. RNA polymerase 1 synthesizes all the rRNAs from the duplicated sets of ribosomal genes. RNA polymerase 2 is situated in the nucleus and transcribes protein-coding nuclear pre-mRNAs. Eukaryotic pre-mRNAs undergo extensive processing after transcription, but before translation, and we'll look at that also. The term mRNA refers to mature processed molecules ready for translation in the discussion of transcription in eukaryotes. Finally, you have RNA polymerase 3, which is located in the nucleus, transcribes various structural RNAs, including 5S pre-rRNA, transfer pre-rRNAs, pre-tRNAs, small nuclear pre-RNAs. tRNAs are the big deal here because tRNAs are crucial for translation. They act as adapter molecules between the mRNA template and the growing polypeptide chain. Small nuclear rRNAs are also important because they are used to splice pre-mRNAs and regulate transcription factors. To identify which polymerase transcribes a gene, scientists can use alpha-amanitin, which is a toxin produced by certain mushrooms like the fly agaric toadstool. Alpha-amanitin affects the 3 polymerases differently. So RNA polymerase 1 is not affected at all. RNA polymerase 3 is moderately sensitive, whereas RNA polymerase 2 is highly sensitive. In fact, it will stop transcription entirely. Since RNA polymerase 2 transcribes the majority of genes, we're going to focus on it for our discussions moving ahead, talking about eukaryotic transcription factors and promoters, okay? So eukaryotic promoters, again, are more complex than prokaryotic promoters, but they share a similar sequence to the upstream 10 sequence found in prokaryotes. In eukaryotes, this sequence is called the TATA box, T-A-T-A box. It has the consensus sequence T-A-T-A-A-A on the coding strand, and it's located 25 to 35 bases upstream from the initiation, the +1 site. So figure 1510 here is showing the eukaryotic promoters and the TATA box, right? Here's the promoter, and then on this end you have the TATAAAA sequence, and this is 30 upstream from the +1 site. Then you have your transcription factors that are binding. So instead of having a single sigma factor like in prokaryotes, eukaryotes have a complex system of transcription factors that recruit the RNA polymerase II to protein coding genes. The factors, known as basal transcription factors, have names starting with TFII, which stands for transcript factor polymerase II, followed by a letter A through J. The core complex is TFIID, which includes a TATA binding protein. Other transcription factors join in a systematic manner, stabilizing the pre-initiation complex and aiding in their recruitment of RNA polymerase II. Some eukaryotic promoters may also contain a conserved CAAT box or cat box, and the full sequence for that is GGCCAATCT, and that's usually around the upstream 80 mark. And then further upstream you have a GC rich box, so the GGCG or octamer boxes ATTTGCAT. Listen, you don't need to know all of these, but just know that there are several of them, and they all have very specific binding sites and specific roles and helping the RNA polymerase II initiate transcription. And know that it's much more complex to initiate transcription in eukaryotes than it is in prokaryotes. Promoter structures for RNA polymerase I and III involve slightly less complex sets of transcription factors compared to RNA polymerase II, but the general concept is the same. Genes transcribed by RNA polymerase I have two GC-rich promoter sequences, and then genes transcribed by RNA polymerase III have upstream or internal promoters within the genes themselves. Following the formation of the pre-initiation complex, the polymerase is released from the other transcription factors, allowing elongation to proceed, just like it did in prokaryotes. RNA polymerase II, responsible for transcribing the majority of eukaryotic genes, synthesizes pre-mRNA in the 5' to 3' direction, just like it did in prokaryotes. Elongation in eukaryotes encounters the challenge, though, of DNA packaging. So during non-dividing phases, eukaryotic genes are tightly packed as chromatin with DNA wound around histone proteins, right? That we remember the nucleosomes. So to proceed with elongation, the transcription machinery relies on the FACT protein complex. FACT helps to remove histones. It takes them off the DNA template as the polymerase advances. So this allows for the synthesis to continue. And once the pre-mRNA is totally synthesized, the FACT restores to the histones to recreate the nucleosomes. Termination differs among the different RNA polymerases. RNA polymerase II elongates beyond the gene by about 1 to 2,000 nucleotides in eukaryotes, and then the pre-mRNA tail is later removed by mRNA processing. We'll get to that in a second. RNA polymerases 1 and 3 require termination signals. RNA polymerase I recognizes an 18 nucleotide sequence for termination, while RNA polymerase III uses an mRNA hairpin, similar to what we saw in the prokaryotic row-independent termination. After transcription is finished, the eukaryotic mRNAs undergo various processing steps before translation. In mRNA processing, the protein coding sequences are interrupted by non-coding introns that must be removed. We'll talk about that in just a moment. Figure 1511 is actually showing the 5' capping and creation of the 3' poly A tail. So both of these steps are done in order to protect the mRNA as it's leaving the nucleus and going into the cytoplasm. So we have a 5' cap that is added to it. It's a 7-methyl guanosine cap. It's methylated. And then then the poly A tail is added after the stop codon. So it just helps to prevent these important bits in here. So you have an exon here, exon here, exon here, and then this is an intron. The exons are the things that are coding for genes. The introns don't code for anything. So we put this poly A tail here to protect the exons. And then the very last step, like I was saying before, is we've got to remove the introns. So this is how that's done.
Eukaryotic cells must process the RNA product of transcription to form a mature, translatable messenger RNA. The first step in RNA processing is capping, the addition of a special nucleotide to the 5' end. The second step is polyadenylation, the addition of a long tail of A's at the 3' end. A eukaryotic gene typically consists of both exons, regions that encode parts of the protein product, and introns, regions that are not represented in the product. The introns are transcribed along with the exons, but they are spliced out as the third step in eukaryotic RNA processing. Ribonucleoprotein assemblies called spliceosomes do the splicing. The result of 5' capping, 3' polyadenylation, and RNA splicing is an mRNA ready to be translated.
Right, so as you saw in the video, you have pre-mRNA editing or splicing. This is where the introns are removed and the exons are joined together. So this process ensures accuracy and precision. You have specific nucleotides that mark the intron boundaries, and then splicing is facilitated by spliceosomes, which are complexes of proteins in RNA molecules.
The mature transcript for many genes is encoded in a discontinuous manner as a series of discrete exons. Exons are separated from each other along the DNA strand by non-coding introns. mRNAs, RNAs, and tRNAs can all contain these encoded introns, which must be removed from precursor RNAs to produce functional RNAs. Most introns do not themselves contain genes and are simply removed from the precursor RNAs and degraded. There are a number of different classes of intron. Some are excised from the primary transcript by proteins, some by ribonucleoproteins, and some excise themselves. Yet all use a series of trans-esterification reactions to accomplish the task. In all splicing reactions, the two exons that surround an intron are not directly joined to one another in a single step. Instead, a two-step reaction occurs in which the intron is first detached from exon 1, freeing this exon to react with exon 2. Both of these reaction steps are transesterifications in which a single phosphodiester bond between two nucleotides is broken and replaced with an energetically equivalent phosphodiester bond without net input of energy. We will now focus on the splicing of eukaryotic mRNAs. Introns can account for as much as 90% of the length of a typical precursor mRNA. Splicing of eukaryotic mRNA is catalyzed by the spliceosome, a large ribonucleoprotein machine. The spliceosome is composed of 5 individual small nuclear ribonucleoproteins, pronounced SNRPs, which comprise both RNA and protein components. These SNRPs are U1, U2, U4, U5, and U6. Each one contains an RNA molecule called an snRNA that is usually 100 to 300 nucleotides long, together with several protein factors. The snRNAs perform many of the spliceosome's mRNA recognition events by forming Watson-Crick base pairs with the precursor mRNA and with one another. The protein factors recognize specific sequences in the mRNA or help to promote conformational rearrangements in the spliceosome that are required for the splicing reaction to progress. Before splicing can occur, the spliceosome must identify the splice sites, the sites at which exons are separated from the neighboring introns and at which two exons will be subsequently attached. The spliceosome identifies splice sites in part by recognizing extremely short sequence motifs found in each pre-mRNA. These motifs are a GU sequence at the 5' end of the intron and an AG sequence at the 3' end of the intron. In addition to these sequences, a branch point nucleotide within the intron and a polypyrimidine tract upstream of the 3' splice site are key elements that are recognized by the splicing machinery. We also understand that there are general features of exonic and intronic sequences which result in the tendency of certain proteins, like the SR proteins, to bind to exons and other proteins, like the HNRNP proteins, to bind introns. These differences also help to define exons and introns. The first step in the splicing reaction is the recognition of the 5' splice site by the U1 SNRP. Other splice site consensus sequences are recognized by non-SNRP factors. The branch point sequence is recognized by the branch point binding protein, BBP, and the polypyrimidine tract and 3' splice site are bound by two specific protein components of a splicing complex referred to as U2 auxiliary factors, or U2AFs. U2AF65 binds to the polypyrimidine tract, and U2AF35 binds to the 3' splice site. Following the binding of these initial components, the remainder of the splicing apparatus assembles around them, in some cases displacing some of the previously bound components. Next, the precursor mRNA undergoes a structural rearrangement and the first transesterification step takes place. During this step, the 2' OH of an adenosine residue located within the intron attacks the exon 1 intron boundary, detaching the intron from the exon and producing a branched intron structure called a lariat. Other rearrangements in the spliceosome then bring together the newly freed exon 1 and the intron exon 2 junction. The terminal 3' OH of the newly released exon attacks the intron exon 2 junction. This second transesterification reaction completes exon ligation or splicing, releasing the lariat intron. While the molecular details of the individual steps are not fully understood, it's clear that the spliceosome is a dynamic ribonucleoprotein machine, and that conformational changes in the RNA components themselves are central to the spliceosome's function.
tRNA and rRNA are very important structural components in protein synthesis, but they do not get translated themselves. Pre-rRNAs are transcribed, processed, and assembled into ribosomes in the nucleolus. Pre-tRNAs are transcribed, processed in the nucleus, and then released into the cytoplasm for protein synthesis. Both eukaryotic and prokaryotic tRNAs and rRNAs are initially transcribed as long precursor molecules spanning multiple RNAs. Enzymes cleave these precursors into subunits corresponding to each structural RNA. Methylation occurs in the pre-rRNAs and pre-tRNAs for stability, and subunit excision also takes place in eukaryotic pre-RNAs destined to become tRNAs or rRNAs. Mature RNAs constitute about 50% of ribosomes, serving structural, catalytic, and binding functions. And mature tRNAs adopt a three-dimensional structure with base pairing and intramolecular hydrogen bonding. And they are the ones that are responsible for positioning the amino acid binding site and the anticodon for mRNA codon interaction. Figure 1514 is showing a space-filling model of a tRNA molecule that adds the amino acid phenylalanine to a growing polypeptide chain. So this is just an example here. You have the amino acid up here, and then you have the tRNA, and there's a codon, an anticodon, down here at the bottom. The anticodon AAG binds to the codon UUC on the mRNA, and then the amino acid polyphenylalanine is attached to the other end of the tRNA, and that gets attached to the amino acid that came before it. Don't worry, we're going to look at all of this in the video in just a moment. Protein synthesis is energetically demanding, but it is crucial for cellular function. So translation involves decoding mRNA into a polypeptide product, and then the amino acids are linked together by peptide bonds, forming polypeptides ranging from 50 to over 1,000 residues. Each amino acid has an amino group and a carboxyl group. The peptide bonds are catalyzed by ribosomes, generating water as a byproduct. Translation involves multiple components, including mRNA, ribosomes, tRNAs, and enzymatic factors. The specific composition may vary, but the overall structures and functions of the protein synthesis machinery are similar across different organisms. Ribosomes, essential for translation, require energy to be built, and they consist of rRNAs and polypeptides. Eukaryotes have a specialized nucleolus for rRNA synthesis and assembly. Ribosomes are found in the cytoplasm, rough ER, mitochondria, and chloroplasts. They dissociate into large and small subunits and reassociate during translation initiation. Prokaryotic ribosomes are 70S, while mammalian ribosomes are 80S. Multiple ribosomes can simultaneously translate in mRNA, creating a polysome. tRNAs are transcribed by RNA polymerase III. We saw that already. And again, they're structural. The cytoplasm contains 40 to 60 types of tRNAs, and they act as adapter molecules carrying a specific amino acid and recognizing mRNA codons. Aminoacetyl tRNAs bind to the ribosome, adding amino acids to the growing polypeptide chain. tRNAs decode RNA language into protein language, with each tRNA anticodon matching specific mRNA codons. Finally, the aminoacetyl tRNA synthase. So after the pre-tRNA synthesis, amino acids are attached to tRNAs in a process called tRNA charging. Aminoacetyl tRNA synthases, that's a hard one to say, specific to each amino acid catalyze this reaction. ATP is hydrolyzed, forming a high-energy bond between an amino acid and AMP. The activated amino acid is transferred to the tRNA, releasing AMP, and this energy-rich bond drives peptide bond formation. And each tRNA is named after its amino acid. Figure 1515 is showing how a peptide bond links the carboxyl end of one amino acid with the amino end of another. Carboxyl amino. And it produces a water molecule in the process. These two are binding here, CO and H, CO and H, and then you end up with a OH instead of a double bonded O, and you still have this H here, so this forms water. Protein synthesis involves initiation, elongation, and termination. It's similar to prokaryotes with some differences. So let's look at translation, and we'll use again E. coli as a representative. So translation begins with an initiation complex, the 30S ribosome mRNA template, initiation factors, and initiator tRNA. In E. coli, the mRNA's Shine-Dalgarno sequence interacts with ribosomal RNA, positioning the 30S subunit. GTP provides energy. The initiator tRNA is carrying formulated methionine, and it binds to the start codon, the AUG. After forming the initiation complex, the 30S and 50S subunits join. Eukaryotes have a similar complex with mRNA, the 40S subunit, eukaryotic IFs, and nucleoside triphosphates. The charged initiator tRNA in eukaryotes lacks formulation but can bind initiation factors. In eukaryotes, the initiation complex recognizes the mRNA's 7-methylguanosine cap. The cap binding protein and initiation factors assist the ribosome's movement toward the cap. The complex scans the mRNA to find the AUG start codon, and once it finds it, the other proteins and the cap binding protein dissociate, and the 60S subunit binds to the complex, completing translation initiation. During elongation, the ribosome has 3 compartments: the A site, P site, and E site. The A site binds incoming charged aminoacyl tRNAs. The P site holds tRNAs carrying amino acids linked to growing polypeptide chains, and the E site releases dissociated tRNAs. The mRNA template guides tRNA binding during elongation. As the ribosome moves along the mRNA, each codon matches with its specific tRNA anticodon, and this ensures accurate binding and prevents random interactions. Elongation proceeds by sequentially adding amino acids to the polypeptide chain. The ribosome advances 3 bases in the 3' direction, moving tRNAs from A to P says elongation factors provide energy through GTP hydrolysis, facilitating tRNA binding and translocation. The peptide bonds form between amino acids in the A and P sites, and the energy comes from the high energy bond linking the amino acids to its tRNA. After peptide bond formation, the A site tRNA moves to the P site, and the empty P site tRNA is expelled from the ribosome. Termination occurs when a nonsense codon is encountered, and then release factors resembling tRNAs recognize these codons in the A site, and they instruct peptidyl transferase to add a water molecule releasing the protein. The ribosomal subunits dissociate from the mRNA, and they're ready for another round of translation. After completing translation, the mRNA is degraded for nucleotide reuse in future reactions. Figure 15.16 is showing the large and small ribosomal subunit and the 3 tRNA binding sites of a ribosome, right? So during translation, the charged tRNAs enter the acceptor site, the A site, and the anticodon on the tRNA pairs with the mRNA codon. The incoming amino acid forms a peptide bond with the growing polypeptide chain, and the ribosome moves 3 nucleotides forward. This movement transfers the tRNA to the P site and allows the empty tRNA to exit at the E site, the exit site. Figure 15.18, again you can see the E, P, and A site. This is still the ribosomal large and small subunit. And this figure is showing the initiator tRNA anticodon recognizing a start codon on the mRNA that is bound to the small ribosomal subunit. So this is a tRNA, then we have a start codon here on the mRNA strand that is bound to the small ribosomal subunit. As the mRNA moves relative to the ribosome, right, so you have the small and large ribosomal subunit will combine, like you see here, and then as the mRNA moves relative to the ribosome, the successive tRNAs move through the ribosome and the polypeptide chain is formed. So here we see the growing polypeptide chain. Entry of a release factor into the A site terminates translation and the components dissociate. There are more figures illustrating all of these steps in the book, but I think this is a really good time for a video. I think you probably agree.
In protein synthesis, we can look at 2 major steps. One is transcription and the other is translation. Transcription has a C in it and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. Now, transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You could see on a codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA, so the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported. This can all vary based on the protein structure and function.
During and after translation, amino acids can undergo chemical modifications, signal sequences can be added, proteins can fold into three-dimensional structures through intramolecular interactions. Signal sequences act like train tickets for proteins, directing them to specific cellular compartments. Signal recognition proteins are the conductors of the train, so they recognize the sequences. And then once proteins reach their destinations, the signal sequences are usually removed. While many proteins fold on their own, some require chaperones to prevent aggregation during the complex folding process. And even with the correct mRNA, abnormal temperature or pH conditions can disrupt proper folding, which can lead to dysfunctional proteins. So this is showing how the proteins are targeted for secretion. So if a protein has a specific signal at the N-terminus that's recognized by the signal recognition particle, the ribosome will be targeted to the translocation channel in the ER membrane, and the protein will go from the ribosome across the ER membrane and into the lumen. And from there, it can be secreted from the cell via vesicle trafficking. There are over 20,000 different proteins in our bodies. So, how do proteins have so much functional diversity when they're based on only 20 amino acids? And the answer lies in the complexity of the structure. So, you have 4 different types of protein structures. We're going to go through all 4 of them. The primary structure consists of the amino acid sequence, and even at this level, there's a substantial biochemical complexity, including the acidity, size, hydrophobicity, and charge that is conferred by different amino acids. Regarding acidity, there's 3 types of amino acids. You have neutral, like alanine, acidic, like aspartic acid, and basic, like arginine. The secondary structure consists of small-scale 3D structures caused by the interactions between the amide groups. The major classes are alpha helices and beta sheets. Alpha helices are right-handed coiled structures. The variable groups are facing outward, and the structure is held together by hydrogen bonds between the amino acids. For example, myoglobin is a protein with multiple alpha helices, right? And then beta sheets are pleated structures that are formed by interactions of the NH and CO groups of neighboring strings of amino acids. So fibroin, a major component of silk, is almost entirely made of beta sheets. Tertiary structure is the large-scale 3D formation of the protein caused by numerous interactions between the side chains of the different amino acids, the interactions between alpha helices and beta sheets, and then quaternary structure is the arrangement of several protein subunits all together in a complex. Examples would be antibodies and hemoglobin. Now, besides these 4 levels of structure, proteins can be further altered for function by post-translational modifications. Please watch this short video that shows the processes involved in protein folding and the different types of protein structure.
Proteins are made up of folded polypeptide chains, which are composed of 20 different amino acids, each with different chemical properties, that are linked to each other via covalent peptide bonds. The sequence of atoms repeating to form the peptide bonds is called the polypeptide backbone. The side chain of each amino acid, which is the part that makes it different from the other 19 amino acids, can be either polar or nonpolar. The polypeptide chain can fold in many different ways called conformations. Conformations are the spatial arrangement of atoms that can result from movement of atoms without breaking of bonds. Protein structure can be described in terms of 4 levels of organization: primary, secondary, tertiary, and quaternary structure. The primary structure is simply the amino acid sequence of the polypeptide chain, and it is important to remember that this sequence contains all the information necessary for the higher orders of structure. The secondary structure is formed by hydrogen bond interactions of adjacent amino acids. Large numbers of such local interactions form alpha helices and beta-pleated sheets. The tertiary structure is a more compact 3-dimensional shape. Large proteins often consist of several protein domains, which are distinct structural units that fold somewhat independently from one another. Quaternary structure is found in those proteins that have 2 or more interacting polypeptide chains, which are then termed subunits. Secondary structures include alpha helices and beta sheets. An alpha helix occurs where a polypeptide chain coils like a spring, with 1 turn every 3.6 amino acids. In alpha helices, the NH of a peptide bond is hydrogen bonded to the C double bond O of another peptide bond, which is 1 coil up in the helix structure. Note also that all the NH groups point in one direction, towards the N terminus, and all the C double bond O groups point in the opposite direction, towards the C terminus, and this is what gives the polypeptide chain polarity. The C terminus is partially positively charged, while the N terminus is partially negatively charged. Beta sheets can form parallel chains, which are made from neighboring chains running in the same direction, or antiparallel chains, which are made from a polypeptide chain that folds back and forth on itself so that nearby sections run in opposite directions. While hydrogen bonds in an alpha helix are intrastrand, hydrogen bonds in beta sheets are interstrand. Tertiary structure formation can be nucleated by the pattern of polar and nonpolar amino acids in a polypeptide chain, which plays a central role in determining the protein's final conformation. This is because hydrophobic molecules, such as the nonpolar side chains of certain amino acids, are entropically driven together in an aqueous environment. This limits their disruption of the hydrogen bonding of surrounding water molecules. As a result, nonpolar amino acids tend to be found predominantly in the interior of proteins. Meanwhile, polar amino acids face the outside of the protein, forming hydrogen bonds with one another and with the water molecules around the protein. Those polar amino acids that are on the inside of the protein bond with one another or with the polypeptide backbone. It is thought that protein folding happens roughly along the following lines. Secondary structures form first. Hydrophobic collapse, during which nonpolar amino acids aggregate, happens next. Long-range interactions between secondary structures cause further folding to occur. Throughout this process, there may be one or more intermediate states, such as what has been termed a molten globule. As a final note, there are three basic classes of proteins, which are distinguished based on shape and solubility: globular, fibrous, and membrane proteins. Globular proteins are spherical in shape, with as little surface area per volume as possible. These proteins are marginally stable, and this marginal stability facilitates motion, which in turn enables function. Hydrophilic amino acids occupy this small surface area, making these proteins highly soluble in water. Fibrous amino acids are simple linear structures which have structural roles and are insoluble. Lastly, membrane proteins are associated with cell membranes.
This concludes the Lesson 5 material. Thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics and leave your questions for me in the comments below.